Okay. Okay, so today I'm going to talk a little bit about cancer and the role of genes in cancer progression and promotion. And then I'm going to try and link in some molecular techniques and some MCQ questions that might be useful for the molecular techniques and the bits of the curriculum that I wasn't quite sure where else to stick in my presentation. I don't really know how much I'm going to be telling you stuff you already know, but anyway, cancer is effectively a disease of cell proliferation. So it's when your cells go out of control. So it can be in any cell of your body, and there are two key dysfunctions: defective cell proliferation, where cells are growing too fast, and defective cellular differentiation. And this is because a cancer wants to be immortal. So it wants to lose its identity because it doesn't want to be differentiated, because that gives it mortality. It wants to be immortal, live forever. And then, so that's why cancer cells look more like, they want to revert more to embryonic stem cells, because they can live forever then. And so with cancer, it's just that balance between normal growth. So in normal cell cycle, our cells are cycling all the time. We have this balance of growth promotion and growth restriction. And this is where the role of the tumor suppressor and the oncogenes come in. And in tumor cells, this balance is disrupted. So you get continuous proliferation. So very much like if you have cell cycle, you've got like brake and an accelerator. And if within cancer, someone's cut your brake cables. So there's no braking on cell cycle. So cells cycle out of control. And so this can happen by multiple different mechanisms. You can have mutations in genes that are responsible for controlling cell cycle and controlling cellular proliferation. And these may have an important role in the birth of cells or in a role in cell death, so a role in apoptosis. They also can have a cancer that arises from a mutation in a gene that has an important role in the stability of the whole genome. So DNA mismatch repair genes are important for checking that when DNA replicates, that no mistakes are made. And obviously, if you have mistakes in these genes, then they're going to allow mistakes to accumulate. And so cancer comes to us because of many different reasons. Inherited susceptibility. So typically, if someone presents with cancer at a younger age, so typically under 60, maybe under 50, then it's likely that they've probably got an inherited susceptibility to that cancer. They've set off at a position whereby, and then one of these genes that has an important role may already be mutated. And often then they acquire new mutations. We, as a function of living, we acquire mutations because of all the kind of crap we throw at ourselves. And so cancer is effectively a disease of old age. So as you get older, you're more likely to accumulate more mutations. Thus, you're more likely to get cancer because of that environmental interaction. And for different cancers, they're all different. So it's not one disease effectively. It's multiple diseases, different numbers of mutations needed for different cancers to actually go from a mutated origin mutated cell with a mutation in a driver gene through to a cancer that is at a large stage and potentially then metastasizing. So we couldn't often talk about cancer as a genetic disease, although there is that genetic risk. So some people will have an inherited genetic risk. So you may inherit a mutation in a BRCA gene or something, or in something like Leigh-Ferroni syndrome and a P53 gene, where in those families, people will present with cancer at a much younger age. But most cancers have mutations in somatic cells. So again, referring to the cells of your body that you don't pass on to your kids. Estimated only about 1% of cancers are actually germline. And so the sort of research that we do, we're looking at the genetics of the cancer rather than the genetics of the individual. So we often, the kind of research that I do anyway, we look kind of characterizing the cancer and looking at it as a separate entity than the genetics of the person, because as a cancer evolves, it acquires more mutations that aren't occurring in the rest of that person's body. So they're somatic mutations. So in most cancers, an inherited mutation alone is not sufficient. You need to get another one. And this kind of refers to Knudson's two hits hypothesis, and I'll talk about that in a minute. Key features about tumors, differentiation, anaplasia. They don't become differentiated. It doesn't look like the cell that it's meant to look like. You get genetic instability, you get multiple mitoses, abnormal numbers of chromosomes in the cells. And then basically cancer cells are like that neighbor everybody hates. They become antisocial. So they have these key factors that normally cells are nice and they're nice and compliant, and they do what they're told, and they listen to signals from all the other cells around them, which means they don't grow out of control. As a cell mutates and has a mutation, it becomes more antisocial, and it doesn't listen to those signals. It doesn't obey the rules of being a cell, and it starts then to evolve into a cancer. And so key factors are it becomes independent of needing growth factor signaling. It doesn't have an awareness of, normally cells can know that other cells are nearby, so it doesn't have an awareness of cell density. It will still grow irrespective of whether there's enough cells there. It has impaired cohesiveness and adhesion, so it's not sticking, or it's not sticking to its cell membrane. It doesn't talk to the other cells, and then it becomes immortal. So all of these kind of are traits that are kind of like, when I talk to kids about it, I say these are like features of a supervillain that a cancer requires to become immortal, so that it can survive in the body and then persist and grow, and then eventually develop its own vascularization, its own way of feeding itself, and it can grow and then metastasize. Key feature for cancer, so again, potentially could be a question a bit about, this is how they could chuck in cell cycle, I guess, revising. So again, mitosis is key with cancer. So mitosis, if you remember, it's like the whole process of cell replication, where you've got prophase, metaphase, anaphase, telophase. So those are the phases of whereby the chromosomes line up in the middle of the cell, and they pull apart, and then the cell divides effectively. And this is all important, obviously, in cellular replication. And so this is a diagram here of cell cycle, and showing some of it, its control. And this is how the genes like P53 come into play, which is a major tumor suppressor gene. It has a role in breaking the cell, acting as a braking mechanism at the cell at different points to enable that it allows sufficient time for the integrity of the DNA to be checked. If you don't have this braking mechanism, and you don't have a way of the cell checking itself, then obviously you get more mutations acquire as the cell goes through a cycle, and more mutations are likely to occur in genes that have some important function, and so which will then allow other mutations to acquire and enable this cell to get out of control. So the way in which cancer develops in terms of relating it to the genetics, so obviously you've got initiation, promotion, progression. And so you start off here in this, if you look at the tissue here, you've got your first mutation, which would be like cancer driver mutation, genetically alters this little cell, and it becomes different from its neighbors. It's no longer the same. So it's had some sort of mutation that gives it an advantage over the local population of cells. And over time, this cell will then proliferate at an increased rate. So then you kind of get hyperplasia here. It kind of still looks a bit like its descendants, but it's growing too fast. And after, could be years, could be, I don't know, it depends on what the cancer is, could be a long period of time. One of these hyperplasic cells will going to acquire another mutation, and then a further mutation again. And each mutation gives this population of cells an advantage in growth over its neighbors. And then you result in getting dysplasia. And when you've got dysplasia, the cells will start to appear abnormal. The tissue is then looking dysplasia. It looks different. It doesn't look like how it should do. The shape and orientation of the cell is wrong. And again, more mutations can occur and enable this population of cells to grow rapidly, mutate, acquire other characteristics. In terms of how that relates to what your body's doing the whole time. So if you're relating it to an environmental trigger, you could be, your body's being exposed all the time to procarcinogens, whether these are chemical or physical. And your body's basically designed, so if you go and have a cigarette now, your body's probably basically designed to counteract that. And so although it's something harmful, your body's designed to counteract that. If it doesn't counteract it, then the procarcinogen becomes activated into a carcinogen, and it can damage the DNA. And again, your body's designed to counteract that. If you take a cigarette and you don't have a cigarette and it effectively mutates your DNA, it does damage to the DNA. Again, your body's designed to have programmed cell death, apoptosis. All the DNA repair mechanisms are in place to try to repair that damage. If that doesn't work, then you have an altered cell. So this is where, and this could happen whereby a gene in that piece of DNA could be mutated from being a proto-oncogene to an oncogene. And so then you've got that driver mutation has happened, and that might persist like in that first genetically altered cell for years, potentially years, or other mutations can occur. And sometimes the immune system is able to remove these mutated cells, which is again why a lot of the treatments now are targeted at, biologics are targeted at using the immune system to target cancer cells. Or that cell can remain dormant and may not ever develop into a cancer. But if it does get mutated and develops into a cancer, then you get progression, where then eventually you get uncontrolled cell replication, loss of, as I said, loss of cell specialization, and then invasion of other tissues by the cancerous cell. Just some key differences between benign and malignant. They're pretty obvious. A benign cancer is nicely contained, typically as a fibrous capsule. The cells are more differentiated, has a slower rate of growth, but it's unable to suppress proliferation. Whereas in a malignant cell, it's less differentiated. It grows much more fastly, grows quicker. And there are essentially two categories: solid tumors and the blood cancers. In terms of metastasis, so they might ask you questions about, I guess, so with uveal melanoma, and again, being aware of like particular mutations that are linked with particular phenotypes. So that might be something here, but metastasis is basically spread from a primary site to a secondary site. Typically, this is by lymph or by blood. So it's when you've got a population of cancerous cells and a cell effect just breaks off and then it finds a way. And again, some of these features that the cancer population will acquire will be developing the production of enzymes that allow it to make holes effectively in the blood vessels or in the lymph vessels to allow the cancer to penetrate through and then the little cells to run off and then seed somewhere else. So these tumor cells can secrete enzymes that enable them to get through the membrane and enable the cells to break off. So they have this loss of cell adhesion, but that's one of the other features they acquire, which enables them to break off that population of cells and then go and seed somewhere else. Other factors that the tumor enables it to survive could be the release of angiogenic factors, which allow the tumor to grow its own vasculature as well. With uveal melanoma, it's a particular mutation where there's monosomy 3 is associated with high potential for metastasis. So that may be something that might be worth focusing on. Genes in cancer is therefore really important. So I said before, we can recognize from that small percentage of people that have inherited mutations, we can identify individuals who are at a greater risk, so BRCA1 and BRCA2. So just as put it in context, a BRCA1 or BRCA2 mutation is much more likely to be found in young women with breast cancer. So found in about 33% of women under 29 with breast cancer, and only found in about 2% of women who are age 70 or above with breast cancer. Another reason why it's important to know about some of the genes associated with cancer is that not only does it mean for a diagnosis or identifying a particular cancer type, but also now development of specific treatments that are targeted to work, that are known to work better depending on the behavior of that tumor. So effectively what a lot of research is focusing on now is looking at the molecular fingerprint of a cancer, looking at what particular genes are mutated to understand what is the likely behavior of that tumor, and so to target then a particular therapy that will work or is known to have a much better chance of improving the outcome for the patient. And other factors, when we talked about epigenetics the other week, obviously there are other ways in which genes can become mutated or lose function. It's not just through mutation, it's also through epigenetic factors as well. So again, when we talk about epigenetics, it's the expression of a gene can be silenced without actually mutation occurring, or it can be, or the expression of a gene can be switched on through the removal of methylation, for example. Key differences between cancer and genetic diseases is that cancer is typically mostly caused by somatic mutations, and most other recessive conditions are other genetic conditions caused by germline mutations, and that the cancer is this result of a multi-hit process. So key genes involved in cell division. Genes that suppress cell division, tumor suppressor genes, so pretty obvious. Their role is to control abnormal cell proliferation, and they suppress tumorigenesis. So P53 and retinoblastoma 1 are two really important tumor suppressor genes. Genes that promote cell division, so their normal role is, when it's in their normal function, they're called proto-oncogenes, and their normal role is to promote cell proliferation and differentiation. If a proto-oncogene becomes mutated, it's called then an oncogene because it's then lost that control. It's not doing its job because it's meant to, it's doing the job and it's out of control in terms of proliferating. And typically oncogenes are things like growth factors, transmembrane kinases, signal transducers. So tumor suppressor genes switch off cell proliferation, and they're active during somatic growth, regeneration, and repair. And so it's their balance that is lost in a cancer. So typically you need at least one functional copy of a tumor suppressor gene. So often they're recessive in terms of their link to cancer. So you've got 2 copies. And this is why cancer is a function of old age, because if you've got to get a mutation in both copies of your gene, it's obviously going to take you longer to get a mutation in both copies to knock out the function of that gene. If you inherit a mutation in one of those genes, you're already pushed along that along halfway, which is why you get a cancer earlier. So retinoblastoma, Wilms tumor, neurofibromatosis 1 and 2, VHL, these are all tumor suppressor genes. Typically, carriers will be heterozygous if it's a familial cancer, and then they get an earlier onset cancer when they've acquired another mutation. So this kind of just illustrates what is known as Knudson's 2-hit hypothesis. So in a normal individual who hasn't got hereditary mutation, they've got to get 2 hits at the same position, at the same locus in that gene, in order to inactivate that specific gene to trigger the cancer. So that obviously this chances of this will increase as you get older, but are much less than if you are born with already one mutation, knocking out one of the alleles, and then you've just got to acquire another one. So this is the reason why people with inherited mutations will present with cancer earlier. Oncogenes, as I said, are mutated proto-oncogenes. Their role is to stimulate cell division. They code for proteins involved in cell proliferation, growth factors, growth factor receptors, signal transducers within cytoplasm, and nuclear regulating proteins. If there is a change to an oncogene, the proliferation then becomes uncontrolled. So I think one that comes up, possibly might come up, is neurofibromin. So it's a protein from neurofibromatosis and interacts with oncogenes to dampen growth stimulatory signals. So it's involved in the control of these oncogenes. Apoptosis is an important thing to learn that comes up, and we'll do some questions in a second about apoptosis. So apoptosis is programmed cell death. And it's a process whereby your body, it should be an active process where the body is trying to defend against the cancer. So if it detects a mistake, it should be initiated apoptosis should be initiated to destroy the cell with the mistake if it can't be rectified, so that that cell cannot persist with mutations in it to allow that population to go out of control. There are some key genes or key proteins. And so there's anti-apoptosis proteins, which are the BCL2 family of proteins. So these stop apoptosis. And then there's the pro-apoptosis proteins, which are BAC, BAX, and BIM. And then P53 has a role in activating most of these proteins. And it occurs in all cells except neurons and is irreversible. So once you program something to kill itself, that's it. So apoptosis is common in malignant tumors and can explain some of the slow growth that you see. And Diane and Amanda will be able to identify and show you what apoptotic bodies are more than I can anyway. Okay, so here's some MCQ questions relating to apoptosis, which apoptosis and genes. So you can have a couple of minutes and look at these. They're all pretty much the same kind of thing. And so maybe just have a look, see if you can answer these, and then I'll go through what the answers are. So maybe I'll just kind of pull bits of questions together to try to make something with some sense. So this first question here, we got genes involved in the regulation of apoptosis include all the following except, which one would you pick? Any guess? It's hard because it's just learning stuff, so it doesn't matter if you get it wrong. I'll tell you what the right answer is.
I think most people were writing it in the chat, but everybody was saying E.
Oh, yeah. Yep, it's E. Yeah. So with HLA, basically HLA is the blood type antigen, normally to do with anything to do with rheumatology or autoimmune if you see a HLA come up. So all the rest aren't. So that I would just do it by process of elimination. Obviously, we've talked about these and their role as oncogenes or role in apoptosis, and P53 is a tumor suppressor. Which of the following statements about null mutations is incorrect? So sometimes you get questions like this that seem like double negatives, and so they can often get questions like this that can be confusing. So what would you put for this one? I don't know if I can see that. Oh, yeah, there is a group chat. I can see it now. Yes, so C, I would go with C. So a deletion doesn't always result in a null mutation, not always. So a deletion can be within a gene, so it doesn't always. So I would definitely look for that one. This one, what statement is incorrect? So this is kind of a mean one. Yes, D, well done. Yeah, FAS doesn't inhibit apoptosis. Okay, about this one, which of the following genes is not associated with regulation of apoptosis? Yep, B is correct. So neurofibromin, we just talked about that one. It has a role more in as a like an controlling oncogenes. And this one here, proto-oncogene in normal cells. What would you put for this one? Yeah, A. So a role of a proto-oncogene is to stimulate cell division. Okay, so that's fine. So you can do those ones. Okay, so now I'm going to just move on a bit, talk about environmental links with carcinogens. So obviously we've talked about how there's that whole process in cancer going from initiation to promotion to progression of cancer. And we talk about how the cell gets mutated from a normal cell to a mutant cell to a malignant cell. And then there's a whole lot of carcinogens that are important, and that repeated exposure to carcinogens, which we get as we get older, increase our risk of mutations persisting and not being removed by the body. Specifically relevant, I guess, to when we're talking about the eye. So the major environmental factors with tumors, chemicals. So I got this from the notes that Diane provided. So chemicals, radiation, and viruses. So specific to the eye, chemicals, formaldehyde, apparently an eyelash glue. Radiation, obviously from UV radiation from the sun or for other types of exposure to UV radiation, and in viruses as well. So from getting conjunctival papillomas, there might be causative effect from HPV-16. So there, in terms of environmental causes, it's important to remember that there are both chemical, radiation, and viral. Okay, so I've got some other questions. So cancer genes and just some key genes that that might come up to be aware of. So ones that we definitely look at when we're looking at panels of genes from multiple cancers, GNAC and GNA11 come up. Obviously from retinoblastoma, and knowing that it's a tumor suppressed gene and RB1. So there's a couple of questions here. Which of the following may contribute to causing cancer? In this. So for this question here, all of these are correct. So all of these can cause cancer. So mutation, faulty DNA, and loss of control over telomere length. What about this one? In regards to uveal melanoma, which is not correct. It's another painful double negative question. So for this one here, none of the above is the correct answer, which makes that horrible double negative question. Hopefully you get questions that are written better than that, but all of these are all correct with regards to uveal melanoma. Okay, so I'm going to talk a little bit about some of the technique stuff and trying to pick up on the other bits that were in your curriculum that and try and find some questions just so see what level they are at. So whole genome analysis, there's different ways in which if we're looking for hunting for mutations or hunting for mutations that are linked to a particular condition, we want to do a whole genome-wide search. And then with the different techniques depend on the resolution, a lot on what money you have available, but also on what resolution you want to look for a mutation. So at the lowest resolution, we have a karyotype. So we're looking at the chromosomes. So obviously, if you're looking at low resolution down a microscope, you're looking for quite major changes. So anything that is cytogenetic, so the large deletions, duplications, translocations should all be observed on a karyotype, and obviously the monosomes and trisomes. If we then go to the next higher level of resolution, we look at our cytogenomic arrays. This is hybridization-based techniques, and looking for large areas of deletions and duplications. And then we've got sequencing. So then we're going to go even finer, where we're looking at the level of the nucleotide, and you have different techniques. So there's Sanger sequencing, I think, would come up on your syllabus. So that's the gold standard of sequencing, and that's looking at small areas of sequencing. And we more routinely now, what we use a lot, we're looking at whole exome sequencing or whole genome sequencing using next generation sequencing. And this will enables us to look for specific point mutations. We also have single gene and panel tests. So we use the Illumina panel 50, which looks at sequencing hotspots in 50 oncogenes and tumor suppressor genes. And we run this on a next generation sequencer machine. So the main difference between Sanger sequencing and next generation sequencing. So Sanger sequencing is still like the gold standard for sequencing, but it's maybe used for sequencing a single gene. It can sequence a smaller number of samples and a smaller number of targets. Is used a lot from in terms of sequencing microbes to identify them, and looking at particular fragments, or for looking at microsatellites. And often it's used to confirm something that's been found still on next generation sequencing. So the reason why we use next generation sequencing a lot more now is that it's quicker and we can look at a lot more data. So we can look at more than 100 genes at a time. So it's more cost effective to use next generation sequencing. And you can use smaller amounts of input DNA as well. In terms of putting it into a context of a question, so I found this one. So you can have a go at this one. So we've got a 32-year-old man has Riker syndrome, autosomal dominant condition, anterior segment dysgenesis, and teeth with umbilical anomalies. Mutations in the gene have been found in some patients. He and his wife do not wish to have the condition passed on to their kids. So they have genetic counseling. They wish to pursue genetic prenatal testing or pre-implantation genetic diagnosis. So in order to do that, they need to identify what the mutation is. And so they're saying in this question, arrangements are made for the sequencing of the coding exons of the PITX2 gene. And so which of these do you think is going to be correct? Which one of these statements is correct sounding this? What's it going to tell them when they sequence the coding exons of the PITX2 gene? So the correct answer I would put for this one is D. So it's not going to identify all possible mutations in the gene because it's a sequencing technique, but it's going to look for missense and frameshift mutations. So sometimes it can't detect large deletions, or it can't detect mutations that aren't in the exon. So this is looking at the coding exons. So there might be still stuff in the promoter region or in the intron exon boundaries. So if they're looking at the coding exons, it can look at missense and frameshift mutations in the coding part of the gene. It's not looking at the promoter, and it can't exclude a deletion, even if the sequencing is normal, because it might have a deletion might occur somewhere else. And it can't detect chromosomal rearrangement. So again, that goes back to the karyotyping. So in this case here, the correct answer would be D. So this is just looking at again, just summarizing what I said about the different methods. So again, it's all based on, I guess, cost and resolution. So your low level resolution is karyotyping in FISH. So FISH is where you use DNA probes that are fluorescently labeled to detect complementary DNA sequence. So this requires a knowledge of what you're looking for. So you can't go hunting for a gene using FISH. You can look for a known mutation like a translocation, or look for extra copies of a particular gene or a particular chromosome by using a fluoro probe that will hybridize to that region in the cell preparation. The next level up, so basically cytogenomic arrays will detect more of cases that are unexplained, so there might be a better chance of identifying a mutation. And then like more increased resolution will be a SNP array, and I'll explain what a SNP is in a moment. So this is looking again at the level of the nucleotide. Again, just looking at what you can detect using a karyotype. So it's low resolution, but you can detect trisomies. So it's really good for trisomies. Sometimes you can detect metastasis, and you can't detect things like consanguinity and UPD. But there's a low risk of identifying variants of unknown significance. With microarrays and sequencing, there's a much higher risk of identifying a variant of unknown significance, which can be an issue. Microarrays are good for deletions and duplications. So in terms of whole genome analysis, so this is what like kind of a sequencing setup would look like. They're not very interesting. They kind of look like bread ovens, and you kind of get this data back, which is just like the fluorescently labeled probes on each of the nucleotide bases. If you were looking for a small mutation, then you want to do sequencing because it's, or some sort of genotyping based sequencing, because you're looking at a mutation of a small size. If you're looking for a big mutation, then you can use things like FISH with probes, hybridization techniques, and arrays. If you are looking for a particular gene, it's easier because you're designing primers, so you know where, if you kind of like, you can identify where you're looking. A whole genome approach implies that you don't know what you're looking for, and you're trying to discover what the gene is going to be, and again, or you're trying to characterize something. And this is just showing a cytogenomic array. So basically the array is, again, it's a hybridization based technique whereby you hybridize the control DNA and patient DNA, and you look for a loss or a gain of a signal to indicate whether there's a duplication or a deletion in a particular area. And again, this is a targeted, because you're targeting lots of little probes to lots of known areas of the genome. And so you then identify which area there are areas of imbalance. With next generation sequencing, basically you chop up the whole genome into lots of tiny little reads. They're called reads, and these sequences are read, and then you have to then align them all back to what is the template of the human genome. Some of the issues that we have with this is that there's obviously no control human genome, so we have an amalgamation of what we use as the control human genome, but everybody is different, so there's always that element of variation from what we map sequence back to. And also our reference genome still has some gaps, so sometimes it can be hard to align particular sequences. Obviously there are biases with different ethnic populations and what the standard population you're mapping sequences back to, whether or not that's representative of all humans or a subset, or is more biased to certain humans than others. If you're doing whole genome analysis, obviously we are incredibly different from one another. So each person, as an idea, a typical variation you'd have from a reference genome could be you'd have between 5 to 10 million single nucleotide variants. And these can be within coding parts of genes, and also in non-coding parts, and then they can be synonymous. They can be synonymous and non-synonymous mutations, so they can change amino acids between people, and they couldn't be non-disease causing. They can just be variants. There's also a huge number of in-frame and frame shift insertions and deletions, and also copy number of variants. So each person does vary a lot from what a standard human genome is. So there is that difficulty in interpreting data when you're looking for genes and actually knowing what is a significant mutation and what is just a chance variant that you're seeing. We try to reduce cost and to target it into genes. People might do exome sequencing. So again, you're just looking at those exomes. Remember, with a gene structure, you've got introns and exons. The exons are the coding part of the gene. So by sorry, it's a question there. Synonymous is when but you've answered it correctly, so that's fine. With exome sequencing, you're just focusing on the coding part of the genes. So you're obviously you're reducing your cost and reducing your coverage, but you're focusing on the bit where it's likely to have greater impact. So it's likely to have, this is what we talked about when we talked about mutations. If you get a mutation in an exon, and if it's a non-synonymous mutation, or it's a missense mutation or a frame shift mutation where you're changing the code and you're changing the amino acid, then it's likely to have a greater impact. And sometimes there can be particular, if it's a known gene that's linked with a particular condition, but they want to identify a particular mutation in a family that might be a new mutation, then they might do exome sequencing for a particular gene on its own. It might not be every exome of every gene. It might be a targeted exon sequencing for that particular gene. Key issues with sequencing and this kind of technology is that unanticipated pathogenic variants. So if you're looking at whole genome data, then you're going to find a lot of information that you're not looking for. So that can be a difficult complication. So you might find a hereditary cancer predisposition gene when you're looking for something else, if you're looking for a neurodegenerative disease. And so the difficulty here is like, what do you do with this information if you can't act on it? So obviously it's the patient's information, but then once you have this information, you can't take it away. And the consent issues around genomic data are quite important in terms of like how that information is interpreted. And I was talking to one of the genetic counselors here, and there's lots of cases whereby someone had a variant and they thought it was a variant of significance, and they were told that they had a variant that was in a high risk variant for a cancer, and like 10 years later they kind of went back into clinic. They were having regular colonoscopies because it was for bowel cancer, no presentation of cancer, and then 10 years later that variant that was meant to be a significant variant was reclassified as a variant that's not likely a benign variant. So that kind of like sometimes variants can get reclassified. So, but that does have huge implications for a patient if you're living with been told that you've got a variant that's increasing your chance of cancer, and then all of a sudden, like after a period of time, research tells us that it probably isn't, then that can be quite difficult information to manage, and vice versa. So genome data is kind of complex at lots of levels. The technology means there's limitations in terms of you need this huge amount of data that's generated from sequencing. So that there's obviously limitations with data storage and the capacity of the machines that do it. But also interpreting the data we're still not at a point where we can interpret the data with accuracy, though we know that there's a particular mutation that's a higher risk or linked to in some cases, but each person has a different background in which those mutations are operating on effectively. So we're still not at a point, humans are incredibly complex where we can really understand everything. Okay, PCR I think seems to come up on your syllabus. So PCR is basically, it's a way to amplify DNA. So it revolutionized molecular biology because up until PCR, you couldn't really do much with a tiny bit of DNA, but PCR meant you could have a tiny, tiny bit of DNA from a hair follicle or for anything, and you can amplify that piece of DNA to have enough DNA that then you can carry out further analyses. Essentially, so PCR is used to amplify DNA. Basically, you're using a polymerase enzyme. So basically, you're copying what the cell does in terms of replicating DNA, but you're targeting it to a specific area. So typically with PCR, you might want to amplify an area of a gene that you're interested in. And so in order to hone in on that particular section, you use primers. And primers are like flags, and they anneal because they are complementary to the DNA either side of the area that you want to amplify. So they stick on to that part of the DNA, and the enzyme then copies, and it works copying just the DNA that's between those two flag points. And it's through cycles of basically you increase the temperature to separate the double-stranded DNA, and then the primers stick on. So so you've got increased temperature, then annealing is when the primers stick to the DNA. And then you have the polymerase, you get the elongation phase with the polymerase enzyme. So it's an enzyme and it makes a polymer of DNA. So it's a DNA polymerase, and it extends the DNA, copies it by adding in nucleotides through a particular length of time. And then the temperature's dropped again to allow the primers to come off. And then the whole cycle happens again. And what happens is the amount of DNA that you've got and then amplifies exponentially, because each round of amplification, you've got double the amount of DNA that the primers can act on. And so key ingredients in a PCR, you need primers, you need your template DNA. So this is your patient's DNA. It can be like buccal DNA from a swab or blood or whatever it is, but DNA extracted. You need an enzyme, which is your Taq polymerase. So it's a Taq polymerase is used because it operates at higher temperatures, because it's isolated from a bacteria that works at high temperatures. And then you need to add in your fake nucleotides, and it has a buffer. And essentially that's all a PCR is. It's just these ingredients, and it's amplifying that region of interest. So 3 steps in a PCR: denaturation, so separating your double-stranded DNA. So we do this by heating it up to 93 degrees. And then your primers stick onto it. So this is the annealing of primers, and they bind to the DNA either side of the area that you want to be copied. And at this point, the temperature is dropped, and to optimize when the primers can anneal. And that the optimal temperature can vary depending on the composition of the primers. So the primers will have different composition of A, T, C, and G, depending on where you've designed them to amplify. So the temperature for this can be different. And then you have DNA synthesis. This is where the enzyme, the polymerase, uses your nucleotides to copy that piece of DNA. And that normally happens at about 70 degrees. And then it happens again and again. And typically there are about 30 to 35 cycles in a PCR. And then there's a link here that you can watch. There's a million animations you can look online. If you just do animation PCR, there's a link here that you can watch one as well. So I don't think you'll be asked to reproduce what the all these steps, but just recognize what the what the stages are. So Taq DNA polymerase is just an enzyme that it's able to use the nucleotides to elongate the strand from where the primers. It can just stretch them out. Once the primers are there, it can help stretch out that piece of DNA, copying everything between the 2 primers, and the primers act as flags. So I found a whole load of PCR related questions. So I think this is kind of give you an idea of understanding the level maybe of what you need to know. Okay, so this question here, the amount of DNA can be increased more than 10 to the 6 by using. So again, this is just recognizing here that PCR, polymerase chain reaction, is the only technique that you will use to amplify DNA. So if they're talking about increasing the amount of DNA, they're talking about PCR. The other things are just all words. Ligation is sticking, and you use a ligase enzyme. That might happen if you're using a restriction enzyme to cut and insert particular parts of DNA. Restriction endonuclease would be an enzyme that cuts at particular restriction points. Reverse transcriptase is used in the production of cDNA. So all of these things are just words they've stuck in to deter from the fact that they want to say PCR. In this one here, it's got the steps involved in PCR are all correct, except which one? Which one here do you think is the wrong answer? Yeah, B. Yeah, so anything to do with proteins isn't DNA. So as a researcher, I'm very much molecular focused. I don't really care about proteins. I really care about DNA. So I'm not interested in proteins. Typically proteins and antibodies would be, if you're using anything protein related questions, will be like a Western blot. So if they're talking about what technique to take, typically it'll be a Western blot. Southern blot is high. So basically all blots, if you have northern, southern, Western blots, are all hybridization techniques. Again, we don't use them so much now. We use Western blots to identify proteins probably the most. But for information, a southern blot is hybridizing to DNA, a northern blot is hybridizing to RNA, and a Western is hybridizing to proteins. But a PCR is basically it's nothing to do with proteins or antibodies. Okay, this one, regarding primers, which is incorrect? This one might be slightly harder. Yeah, good. B. So B is the correct answer. Again, well, the incorrect answer here. So yeah, primers are all typically about 20 to 30 nucleotides. They occur around that temperature of 50 to 65. They are formed from DNA with short flanking sequences. And if they have more GCs in them, then they typically have a higher melting temperature. So often in the design, we look to make sure that the balance or the location of where you design the primers is optimal for what is going to be easier to create a reaction. PCR, again, just asking what it is. Which one here is true? Yep, C. So you can see, like, once you know a little bit, the rest just seem garbage. So a lot of these other answers don't seem to make sense at all. So yeah, C is the correct answer. And this question here, PCR involves, what would this one be? You can rule this one out. Look, if you're not interested in protein. This one is not true because you need to know where you're amplifying. So you've got to design those flags. So you have to know knowledge of what you're going to amplify. It's amplified way more than 5 times. There is denaturation, but that's not what PCR is. PCR is polymerization. So in this one, I would go with polymerization. It is, you do denature the DNA, but only to get the primers to anneal, and then it's you're making polymers, making lots of copies of the DNA. And there's another one here I found that had a PCR in it. So gene therapy includes all the following except, what would you put for this one? Yep. So basically, these are all types of gene therapy, and that's just like they randomly stuck on PCR amplification. Now, to be a pedant, you probably would use PCR at some point during gene therapy to amplify some sort of DNA, but yeah, effectively, the strategies aren't anything to do with PCR for gene therapy. Okay, you had in your syllabus some of these things. So these are just all looking at DNA polymorphisms. So we don't use these techniques really anymore. SSCP is single strand conformation polymorphism. I did it for my PhD, and it's a nice, pretty way of detecting polymorphisms, but they don't seem to be done anymore. So I don't think you'll get questions on these and DHPRC. If you get something on RFLP, it's a restriction fragment length polymorphism, and it uses hybridization to detect specific DNA restriction fragments in genomic DNA. So this is crucial for DNA fingerprinting and paternity testing. This is where you get those nice long lines of black lines that is unique to an individual, and basically the DNA gets chopped up and probes get used to hybridize to areas of DNA to get this like ladder or pattern, and the DNA gets chopped up using restriction enzymes, which is why it's called RFLP. Different types of polymorphisms, duplicated DNA of distinct lengths in different alleles, that's variable number tandem repeat. If it's just a base pair that differs, then it's a SNP. So a SNP is a polymorphism that just differs at a single nucleotide position. And a microsatellite is a repeat that can be a repeat of 2, 3, or 4 base pairs. So the same bit over and over and over again. LOD scores, like no one uses LOD scores, I don't think, unless you're probably doing animal breeding. But I've just put this in here. So basically, a LOD score is looking at the likelihood that that two genetic locations are in fact linked. So typically if they're closer together, they're likely to be linked because during crossing over, they're unlikely to get separated out. So if genes are closer together, they're much more likely to be linked because they, in crossing over, otherwise, if they're far apart, they'll get separated. In terms of recognizing what they are, a LOD score greater or equal to 3 is normally suggestive of linkage, that these two loci are linked, that they're close together. And if a LOD score is negative, then it's not linked. So I got some questions here. So what does LOD stand for effectively? So that's probably useful to know if they ask you. So LOD just stands for logarithm of the odds. That's what it is. And here you might get a question, which LOD score is proof of linkage? What would you go for here? Yep, C would be 3. I don't think they can ask you anything else on that. I don't think I can tell you any more on LOD scores just because I would never use them. I'm pretty sure that they're not used so much now. A SNP is a single nucleotide polymorphism. So as I said, it's a change of one base pair for another one. There's thousands in the genome. They're often used to identify association with a common condition. So people look as part of a GWAS study. So GWAS is a genome-wide association study. So SNPs are essentially thousands of flags throughout your genome that are known to be frequently different between people. And they're looked then for an association with a particular phenotype of a disease to see whether or not, and it may be that SNP is in a gene or it's close to a gene, close by to a gene that is likely to have an impact on that disease. And SNPs are what I looked at when you have like one of the 23andMe tests, or if you have any of those things. So I had one of those, and they give you, basically you spit in a tube and they do your genotype at 60,000 SNPs throughout the genome. And that's how they tell you all that stuff about like your ancestry and like, you know, whether you're likely to have curly hair, all those kind of things. So a bit about genome-wide studies. You might get asked about genome-wide studies because they were super popular for quite a long time, and people still do a lot of genome-wide association studies. I don't like them because you need huge populations. So as a researcher, they're really hard because you think about the amount of variation you've got between people when you're trying to determine a phenotype and then also a genotype. So to have it statistical significance, you need massive numbers just for an effective population size for a GWAS study. So 20,000 plus people need to be in a GWAS study. You can't do a GWAS study on like 100 people or something. It's meant to be an unbiased way to search the genome and to look for common diseases, or so particularly for common complex diseases. And then people use different GWAS studies. They use case and control, or cases with different degrees of phenotype expression, or they might use familial relationships. What it's doing is effectively it's using genome-wide linkage disequilibrium. So the fact that genes close to these points will be inherited with these points, so that they will be linked to them, to look to see or try to identify an area or that was likely to have an effect to the phenotype you're looking at. The SNP changes, yeah, as I say, are usually signposts. They're not normally pathogenic mutations. They're normally a signpost to say something in this region is likely to be associated with this disease. And researchers like to present this data as something called a Manhattan plot, because it's like a large amount of data. It's just a type of scatter plot. So you might be asked about that. A Manhattan plot is just like a way in which they look at this kind of data. It's called a Manhattan plot because it looks like a Manhattan skyline. It's just like lots and lots of data. Right? Polygenic risk scores are kind of like, people use some of that in clinics. It's not equivalent to a diagnostic test. It's basically looking at a series of variants or a series of SNPs that someone might have and trying to calculate a mathematical risk based on this number of variants that they have, maybe in high risk genes or in areas that are known to be of susceptibility towards a complex trait. It's not routinely used in clinical care, but they may be used increasingly in terms of, alongside for complex conditions like heart disease, obesity, diabetes, things like that, alongside environmental risk factors and also lifestyle risk factors. Some of the cancers are more advanced in using these risk scores. So there's something called Bodicea that's used in breast cancer risk prediction that's quite good. That uses family history and other factors, which is essentially like a polygenic risk score model to determine someone's risk of getting breast cancer. I don't think you'll get asked about CRISPR, but I just thought I'd just say what CRISPR is. So CRISPR is just a mechanism that is now used to do targeted gene therapy. So whereas gene therapy before was kind of hit and miss, you kind of basically, if you were designing gene therapy, you would get the gene or the gene of interest and you would have it in a virus, and then you just randomly stick it in a person and hope that it would work and hope that you get to the right place. The cool thing about CRISPR is you're still doing gene therapy, so it's still gene therapy, but you're making use of the CRISPR-Cas9 enzyme. So this was a system that exists in viruses. It's kind of a neat little system that then targets the gene repair that you want to do, like in PCR with primers. It's like a targeted approach using the primers of the matching gene sequence, so you can go into cell populations and edit a genetic mistake at a specific location. And it's not, there's a lot of interest in this in clinical trial area. A big problem with this is that if you're going in and you're editing genes that you can't, whether or not you can unedit it afterwards. So the Royal Society in New Zealand had like a big discussion thing about what we think about gene editing, and then there's kind of like, you can think about different scenarios, and probably not really relevant for your exam, but there's a whole range of different scenarios where people discuss whether or not CRISPR is relevant. So the targeted gene therapy, so for example to treat a monogenic disease like sickle cell, or to treat cystic fibrosis. So it's kind of like a targeted gene therapy. They seem to be the more acceptable uses of CRISPR. And then there's like also, you know, discussions about whether or not you should correct a hereditary mutation in a BRCA gene, whether or not you can use CRISPR then to activate a gene that will give you better cardiovascular health by activating a mutation that's known to improve circulation or something. And then to the point of where people then discuss about whether or not you can have genetic enhancement, where you could, for example, genetically modify to increase your EPO production. So you want to go and compete in the Olympics or something. So there's a whole range of scenarios of, you know, that we can talk about CRISPR-Cas9 and what is acceptable or not acceptable in terms of what cells you're treating. It targets a specific location, so effectively using a primer sequence. So you design, so you know, you have to know, so basically you have to know the gene that you're trying to correct. So you need to basically have the matching DNA sequence of the bit that you're trying to correct. And then you have this enzyme that comes in and repairs the bit that is wrong and makes it how it should be effectively. So it's a complementary based system, like kind of like a primer system that is matching the bit that you're trying to repair. So you do need to know, with something like CRISPR, you need to know exactly what the gene is you're trying to correct and what you want the right sequence to be. I think that's they're just the scenarios. So I think that's mostly what I was going to talk about. I can look for some more MCQs for you, but I think from the point of view of those aspects of your curriculum, I can't imagine that they would expect you to know in any greater detail than the type of MCQs that I've given you examples of. So knowing that for PCR is for amplification, knowing, you know, that what techniques of hybridization, in knowing effectively just the basics of what PCR does, just is that like that increasing the amount of DNA. I don't think you need to know those things. I wouldn't imagine in great detail. Are there any other questions? Oh yeah, I can try and find you some more Yes, I'll try and find you some more MCQ questions. Yeah, I'll do that. So I can try and look for some that are more focused on the cytogenetic stuff as well that we did in the other weeks. Not sure what else they can ask you about technique stuff. I wouldn't get too hung up about it. They can't give you too many on that, I wouldn't imagine. Okay, so if that's all good, shall I go?
Yeah, so thank you. Oh, thank you, Michelle. I'm just starting my video here. That was really great. Lovely. If you stop sharing your screen, we'll
stop sharing my screen. I'll email. I'll try and find the link. So I'll try and put my presentation up today. But if not, I'll email it to you, Diane, and I'll look for some MCQs this week. I lost the link to the Google Classroom because my computer restarted, but I'll have a look, see if I can find it.
Right.
So team,
I'll be able to give it to you.
Okay, thank you. All right.
If you unmute, you can give Michelle a little round of applause. So unmute and give her applause.
Good luck with your exams. I'm sure you'll be fine. You'll ace them. Bye-bye.
Bye.